I promise you, your blood is way more interesting than anything you remember from high school biology. We all know about A, B, O, and, that little plus or minus sign for RH. But honestly, that's just the cover of the book. Today, we're going to dive into all the hidden chapters. Just take a look at this. This shows the surface of a single red blood cell. And each of those little structures you see, Kell, Duffy, Knops, that's an entire blood group system. Yeah, the ABO and RH systems we all know are in there, but they're really just two landmarks in a massive, bustling city of antigens. And just how massive is that city? Well, beyond ABO and RH, there are more than 20 other recognized blood group systems. And get this, scientists have discovered over 300 different antigens so far. It's an absolutely incredible level of complexity that makes every single one of us biologically unique. So, you're probably asking, okay, if we get by just fine knowing our ABO type, why does all this other stuff even matter? That's a great question. You see, unlike ABO antibodies, which are just kind of naturally there, antibodies to these other groups usually only form after our immune system gets exposed to foreign blood, say through a blood transfusion or even during pregnancy. And that's when things can get really serious. And this brings us to the stakes, because when these other antibodies do form, they can have life or death consequences. This isn't just some academic exercise. This is at the absolute core of transfusion medicine and prenatal care. There are two primary dangers we're talking about. First is something called a hemolytic transfusion reaction, or HTR. That's where your own immune system attacks transfused blood. And second is hemolytic disease of the newborn, HDN, where a mother's antibodies cross the placenta and attack her baby's red blood cells. Both can be incredibly severe, and they are exactly why blood banks screen for these other antibodies so carefully. Okay, so how in the world do we make sense of all these different systems? Well, probably the most practical way is to divide them up based on the temperature at which their antibodies react. I know, it sounds almost too simple, but it's an absolutely crucial distinction that helps predict just how dangerous an antibody might be. So here's the key difference. Cold-reacting antibodies are most active at cooler, room temperatures. Because of that, they're generally less of a concern once they're inside our warm bodies. Warm-reacting antibodies, on the other hand, thrive at our internal body temperature, and that's what makes them far more likely to cause serious problems. All right, let's start with the cold cases. These are the blood groups whose antibodies are usually, but not always, cold-reacting. They're often picked up in lab testing. But a lot of the time, they don't end up causing any trouble for the patient. Here we've got two pretty common cold-reacting groups. Most of the time, Lewis antibodies are totally insignificant. The I system is also usually benign, but it has a bit of a dark side. Its antibody is the culprit behind a nasty condition called cold agglutinin disease. Now, the MNS and P systems show how complicated this can get. Just look at MNS. The antibodies to M and N, typically cold, rarely cause trouble. But the antibodies to S, and U, they're warm, and they can cause severe reactions. It's like a system with a total split personality. And it's the same deal with P. Anti-P1 is common and benign, but anti-P, that is a different beast entirely. Okay, now we're entering the danger zone. We're talking about the warm-reacting antibodies. These are the ones clinicians watch like a hawk because they do all their damage right at body temperature. First up is the Kell system. Anti-K is one of the most common and clinically significant antibodies found, right after the main ABO and RH types. It's a really potent antibody that's well known for causing severe transfusion reactions and hemolytic disease of the newborn. And the Kell system has this fascinating and rare genetic condition linked to it called McLeod syndrome. People with this are actually missing a foundational piece of the Kell system on their red cells. This leads to these abnormally spiky red blood cells and a whole host of other health problems. It's a powerful reminder that these little surface proteins are so much more than just blood types. Next up is the KID system. Its antibodies, anti-JAK-A and anti-JAK-B, are just notoriously tricky. They are absolutely capable of causing severe transfusion reactions, but they have a specific reputation for something else entirely. And this is what makes them so dangerous. KID antibodies have this nasty habit of fading over time, sometimes becoming completely undetectable in pre-transfusion screening. But if that patient later gets blood with the KID antigen, their immune system can launch this rapid, powerful memory response, causing a severe reaction days after the transfusion. They are the true silent danger of the blood bank.
Finally, in our tour of the warm and dangerous, we have the Duffy system. Just like KID, its antibodies can cause delayed transfusion reactions, and anti-Phi-A is also a known cause of hemolytic disease of the newborn. But you know what? The Duffy system has one more incredible secret. This is just amazing. A huge percentage of people of African descent have a specific blood type in this system called Phi-A-B. It means they lack both the main Duffy antigens. Well, it turns out that the Duffy antigen is the exact doorway the malaria parasite Plasmodium vivax uses to invade red blood cells. So no doorway, no invasion. It's a stunning, real-world example of human evolution, where a blood type provides a powerful defense against a deadly disease.
So, after all that, what's the big picture here? What does this all mean? Well, it means your blood type isn't just a letter. It's more like a detailed, completely unique fingerprint. So, what are the key takeaways? First, and this is the big one, blood is so much more complex than we commonly think. Second, understanding this complexity is absolutely critical for patient safety. And third, that simple division between cold and warm antibodies, it's a really powerful tool for predicting risk. You know, this quote from our source material just sums it up perfectly. Your specific combination of these hundreds of antigens is inherited. It's a unique mosaic passed down to you from your parents. It's literally a part of your genetic identity, written on the surface of every single red blood cell. From transfusion safety all the way to protection against malaria, we've seen that our blood holds these incredible stories about our health, our risks, and even our evolutionary past. It really makes you wonder, doesn't it?